Hello, welcome. My name is Jill Barnes. I'm an associate professor in the Department of Kinesiology at the University of Wisconsin-Madison. Before I start my talk, I'd like to thank the organizers for the invitation to this conference to speak to you today and share some of my research. I wish that I could be there in person, and hopefully I'll be able to travel and visit there soon. The title of my talk is Linking Vascular Function and Brain Health. Does exercise play a role? I have nothing to disclose. So I wanted to tell you a little bit about my career trajectory before getting into my research. I started my undergraduate degree at the University of Michigan, where I was planning to study architecture. At the time, I was an avid participant in research studies, and one of the research studies that I participated in was led by Dr. Josephine Kasabubu. This was in the University of Michigan Medical School and Hospital, which is shown here, and it was a very interesting study looking at the effects of exercise on hormone regulation. After I participated in this study, Dr. Kasabubu invited me to join her laboratory to work as an undergraduate research assistant. And so I did that for a while, really fell in love with the area of physiology and exercise physiology, and decided to transition to a kinesiology major, studying movement science. I then worked in the laboratory of Dr. Jeffrey Horowitz at the University of Michigan. He was in the Department of Kinesiology. This is the old kinesiology building shown here, where I did a lot of my work. Dr. Horowitz did his graduate degrees at the University of Texas. So when I was deciding what to do after my undergraduate degree, he encouraged me to go to the University of Texas, and he studied there with Dr. Ed Coyle. So after I graduated from the University of Michigan, I then moved to the University of Texas at Austin. I was very interested in working with Dr. Ed Coyle, but at the time they had recently hired a new assistant professor, Dr. Hirofumi Tanaka. So when I was at the University of Texas at Austin, I joined the laboratory of Dr. Hiro Tanaka. I did my master's degree there as well as my PhD there in exercise physiology. After I completed my PhD, I moved to the Mayo Clinic and I worked with Dr. Michael Joyner. He's an anesthesiologist, but also an exercise physiologist, and his laboratory is in St. Mary's Hospital of the Mayo Clinic. So I spent a number of years here training as a postdoctoral fellow before moving on to my current position. One of the things that really struck me about physiology in general is the idea of form and function. So the form or the structure of an organ or a cell or a system is really linked to the function of that system. And I can't think of a better system to describe that than the cardiovascular system. So here we just have a picture of the cardiovascular system, and you can see the heart and the blood vessels. The other aspect of cardiovascular regulation is actually neural control. The nerve is shown here, and the nerve endings are projecting onto a blood vessel, which are shown here. So this is data from my laboratory that's capturing this nerve activity. The sympathetic nerve activity is shown here. This is one of the branches of the autonomic nervous system, sometimes thought of as the flight or fight response. Each of these bursts correspond to a release of neurotransmitters onto the surface of the vessel. You can also see heart rate and blood pressure. I want to point you to this spot right here. There's a small dip in blood pressure. There's a little bit of the elongation in the R to R interval between those heartbeats, and we can see right after this there's a sympathetic nerve burst. So this really elegantly illustrates the linking of the structure of the cardiovascular system and the neural system with the function. So immediately after this really tall, high amplitude sympathetic nerve burst, then blood pressure starts to come back up to normal. So this is really a concept that has driven me throughout my training and also into my laboratory, is really thinking how the structure of a system informs us of the function, and then how changes in function or adaptations in the function can change the structure of a system. So now I'm at the University of Wisconsin-Madison, and I've been here for a number of years. It's a beautiful campus. If you've ever had the chance to visit along the lakeshore, Here is the medical complex. So this is the Wisconsin School of Medicine, as well as the hospital, and also has the Wisconsin Institute of Medical Research, which we use quite a bit for some of the studies. And some of the studies that I'll show you were the data was captured in one of these research institutes. This is a picture right here. This is one of the windows of my lab. My lab is in the Medical Sciences Center. It's the old hospital of the university, and that's where we conduct the majority of our research. So I wanted to give you an overview of the research interest of the laboratory before diving into our topic today. So I already mentioned that the form and function is something that I am really interested in and really has driven me throughout my career. And I consider that our lab studies neurovascular control of the circulation. So the example of that is what I showed here, where the sympathetic nerve activity, and we have the heart rate and blood pressure, and these are linked. But what I'm going to talk about today, and the work that's been happening in my lab over the last 5 years or so, is really focused on neurovascular control of the circulation, but focusing on cerebral blood flow or brain blood flow. One of the questions that we have is, if we have a brain with healthy amounts of blood flow and able to adequate amounts of blood flow to support normal function, at some point over the aging process or disease process, that blood flow becomes insufficient. So we have not enough blood flow going to the brain, and then that can potentially lead to problems with brain function. We think that this switching or this conversion between a healthy amount of cerebral blood flow to not enough cerebral blood flow is in part linked to the blood vessels. So here I'm showing the blood vessels that are in the brain, but I'm also showing some of the blood vessels that lead to the brain, such as the internal carotid artery. So we think the structure and function of these vessels is very important in determining when a brain function changes from what we consider healthy to what we might consider pathological. So we can look at this a number of ways. We can estimate the function of the vessels in the brain. Here's two concepts. Most of what I'll show you today is in regard to vasoreactivity. I won't show too much data on neurovascular coupling, just in the interest of time. But what this is showing is that we can change the stimulus. We can increase the amount of arterial carbon dioxide, for example, and this initiates a response in the cerebral blood vessels so that cerebral blood flow increases. Similarly, we can give a participant a cognitive test, a short-term cognitive test, maybe over the course of 30 seconds or several minutes. And when they're doing the cognitive test, this is going to require additional cerebral metabolism. There's going to be some work that the brain is doing. And so therefore, there's an increase in cerebral blood flow to meet that metabolic demand. So these are two ways that we could estimate how the cerebral vessels are functioning within the brain in a healthy human. So taken together, we believe these factors that I'm mentioning are linked to the structure of the brain and how the structure of the brain changes over the aging process. And so when we think about aging and we think about disease or dementia, this might eventually manifest as changes in gray matter atrophy, increases in white matter hyperintensities, decreases in white matter integrity. Some of these we're going to talk about today, and as well as decreases in cortical thickness. So if we have an impairment in the cerebral vessels, that's going to affect cerebral blood flow to the brain, and that this might affect brain structure. And these factors that I'm listing here, as well as many others that I won't be talking about today, are associated with impaired cognitive function. So if we can look at the cerebral vessels, understand what's going on there, see how this might be associated with brain structure, and even though most of the work I'm going to show you today is in people who are cognitively normal, eventually, over time, these changes could lead to impaired cognition and impaired cognitive function. So let's talk a little bit more about linking these vascular factors and brain health. So today we're going to talk about vascular structure and function with aging. I'm going to talk about how vascular function might affect the brain. I'm going to talk about cerebral blood flow changes with age, and I'll touch on some differences between men and women. I'll also talk about the potential role of exercise. Then we'll move on to talk about the function of the cerebral vessels, including cerebral vasoreactivity and how that changes with age. We'll also talk about the role of exercise, and then we'll think about some measurement considerations, because the measuring cerebral blood flow in the brain is a challenging task. And then lastly, I'll wrap up with some information on exercise interventions. So let's start off with how vascular function changes with age. So here we have a healthy left ventricle and a healthy aorta. So we have a nice compliant aorta, and then there's also going to be a mismatch in how compliant the aorta is compared to how compliant the branching vessels are. Some of these are going to branch into the carotid arteries, and so they will lead to the brain. So in essence, the aorta and then the carotid arteries are supplying the brain. We can think about the interaction between the left ventricle and the aorta. We can also look at the structure of the vessel wall. So here we have this intimal layer in yellow, the medial layer, and then the adventitia. We could also, in animal models, we can take a cross-section of the aorta and we can stain it to look for the different protein ratios. So for example, in the healthy artery, we can look for collagen expression. And we know that collagen is a structural protein that's important for the vessel wall, but what makes these vessel walls compliant is the elastin protein. So oftentimes we might look at the collagen to elastin ratio. So this is in a healthy situation. What about in older adults, or what about what happens with aging? So you can see here that the left ventricle is slightly enlarged. The diameter within the left ventricle, the dimensions, the internal dimensions are a little bit smaller. We see a widening of the aorta, and so this widening of the aorta means that this aorta is less able to buffer the pulsatile flow that comes from the heart every time the heart beats. The other thing that happens with aging is that we lose this mismatch between the aorta and these branching vessels. So as the aorta becomes less compliant, it's not as good at dampening the pulsatile pressure and flow coming from the heart. The other thing that we see is structural changes in the vascular wall. So this intimal layer is thickening with age. There's increases in the collagen to elastin ratio. And we can also see this if we look in the animal model with an increased collagen expression in older animals compared to younger animals. In both of these cases, the collagen is showing up in the dark red color. So these are some of the changes in the left ventricle and the aorta that occur with age. Let's talk a little bit more about the carotid artery. So when we think about how compliant an artery is, oftentimes if we're thinking about brain health, we're measuring the carotid artery. But in general, the large central arteries are highly compliant. So this means that they're able to dampen the pulsatile flow that comes from the heart. You can see the various layers here. The endothelial cells, that innermost lining, are shown in blue. These red arrows are showing pulse pressure, and as the pulse, as this pressure moves through the artery, this healthy artery is very good at dampening that flow. That's in contrast to what happens with aging or what happens with arterial stiffness. Here we have the pulse pressure, and notice that the pulse pressure is the same throughout the artery here, and there's no buffering of the pulsatile flow or pulse pressure throughout this artery because of the stiffness. How does exercise affect arterial compliance? Before we move on to what's happening in the brain, what is happening to vascular function, what is happening to arterial compliance with exercise? So this is an older study from many years ago. It was done by Hiro Tanaka, my PhD mentor, when he was in Dr. Doug Steele's lab at the University of Colorado. And they focused on the carotid artery in men, and they did a cross-sectional study to look at how arterial compliance of the carotid artery changes with age. So this is the results of their study. So they had all sedentary men. This is showing arterial compliance of young men compared to middle-aged men and then older men. So you can see an age-associated decline or an age-associated difference between the young men and the middle-aged and older men. They also recruited men who were endurance trained. So they regularly performed endurance-based or aerobic-based exercise and had done so for many years. There's still an age-associated change or age-associated difference in carotid arterial compliance, but you can see that this is much less. The difference between the young endurance trained and the middle-aged and older endurance trained is much less when compared with the sedentary men. In addition, these middle-aged and older endurance trained men had significantly higher arterial compliance when compared with the sedentary men. So they followed this study up by taking some of those middle-aged and older sedentary men and enrolling them in an exercise training intervention. This was a 3-month intervention doing aerobic-based exercise. And what they found is that before training, this was the arterial compliance of the carotid artery, and this was 3 months after training. You can see that the carotid arterial compliance is significantly higher after training in these middle-aged and older men. So this would suggest that, at least in terms of arterial compliance, that aerobic-based exercise or endurance-based exercise is beneficial for the health of the carotid artery. This is from a review that was written by Dr. Doug Seals, and this is showing that it's not just any exercise, and it's not just any aerobic exercise, but that the type of exercise may matter when thinking about the adaptations in the vasculature. So this is showing the difference from each of these from untrained controls. So this is the 0 line shown here. So we'll start here with the runners. This is what we often think of when we think of endurance training. So you can see here that the runners, this was the magnitude of difference in carotid arterial compliance in the runners when you compared it to untrained, age-matched controls. Swimmers also have a greater arterial compliance when compared to untrained, and same thing with rowers. But you can see there's just subtle differences based on mode of exercise. What is interesting about this study is that these are all aerobic based, but resistance trained athletes or middle-aged adults who habitually perform high-intensity resistance training actually had lower carotid arterial compliance when compared with untrained controls. So it's not any type of exercise or all exercise that has an effect on the vasculature. The adaptations in the vasculature is dependent on the type of exercise, and later on in this talk we'll discuss a little bit more about intensity. So one question is, if the carotid artery, the arterial compliance of the carotid artery is changing or is different based on types of exercise, and the carotid artery supplies the brain with blood flow, how do these differences in vascular function impact the brain? So this is a model put forth by Dr. Constantino Idicola. He primarily focuses on preclinical work and animal models, but his work has shown a linking between vascular function and brain health. So let me talk a little bit about this slide. So at the bottom we have age, and then here we have a percent of the maximum. And this is following through the age span of 3 phases. We have a latent phase. When thinking about dementia, the latent phase would be a normal, healthy, older adult that has high cognitive function and doesn't have any symptoms of dementia. The next phase is the prodromal phase, which could be compared to mild cognitive impairment. This is where we see some changes in cognition and some symptoms, but they're not considered to have dementia. And then lastly, we have the disease phase that would be dementia, and that dementia might be due to Alzheimer's disease. So next we have this purple line. This is showing neuropathology. So as we age, there's increases in neuropathology that occurs in the brain, and once we reach the disease phase, the neuropathology in the brain is high. This neuropathology could be a mix of things such as changes in the gray matter volumes, could be changes in amyloid or tau if we're thinking about Alzheimer's disease, as well as changes in the white matter. This red line is showing cognitive function. So notice in this prodromal phase, that's where we start to see those declines in cognitive function. This blue line shown here, this is showing vascular regulation. So what we note here is that this time point in the latent phase, when cognition is still high, neuropathology is still low, that's when we start to see some changes in vascular regulation. So this is a particular area that my lab is interested in. It's what a lot of our work focuses on in middle-aged and older adults who are cognitively normal, but when we start to see those subtle changes in vascular regulation. Now, this figure and this model are not saying that vascular regulation is driving these changes in cognition or driving these changes in neuropathology, but it does show that there could be a linking between what's happening in the vasculature and what is happening in the brain when we think about the aging process. So how might vascular regulation affect this, or how might vascular dysfunction, when there is a problem in the vasculature, how might that affect the brain? Well, one thing that this can lead to is disrupted hemodynamics, and I'll show a slide that illustrates that in a moment. These disrupted hemodynamics can be variable and can do a variety of things in different areas of the brain, depending on what the source of this vascular dysfunction is. For example, in some areas of the brain, there may be hypoperfusion that's occurring, meaning there's not enough blood flow to certain areas of the brain. In other cases, there may be too much blood flow going to some areas of the brain, and too much blood flow can affect the capillaries and can cause damage to the capillaries. So we can think, if we think about what happens in stroke, depending on the area of the brain and the type of stroke, some can be a stroke caused by hypoperfusion and some can be caused by hyperperfusion. Together, these will affect neuronal health and can lead to neuronal death if severe enough. In addition, hypoperfusion is going to reduce the amount of energy that can be delivered to the neurons. So thinking about neuronal function and the metabolic activity of those neurons and the cells in the brain, if there's hypoperfusion, they will not be getting enough oxygen and nutrients, and that can also cause problems. In addition, hypoperfusion is also linked to a reduction in clearance. So if we think about Alzheimer's disease, one of the hypotheses about Alzheimer's disease is that the accumulation of amyloid and tau protein that is evident in Alzheimer's brains, that really what's happening is the clearance is the problem. So they're not getting rid of the toxic proteins that are in the brain because of disruptions in the hemodynamics. So collectively, these things can also lead to neuronal death. And then these are all linked to changes in cognitive function. So, for example, if we think of standardized cognitive tests, these things are linked to reductions in executive function, changes in psychomotor speed, as well as reductions in memory. So this disrupted hemodynamics is what we're going to focus on for the next few slides and give a few examples of how this is linked to the brain. So we'll go back to that idea of arterial stiffness and how that's linked to brain health. So here, you may have seen this, a similar slide in a physiology class. This is showing pressure here, and this is showing through the arterial tree. So starting at the left ventricle, when the left ventricle is contracting, we have these large fluctuations in pressure when that's occurring. And then we move through the aorta, the large arteries, the small arteries, and all the way down to the capillaries. So as we mentioned, those large central arteries are really good at buffering the pulsatile flow from the heart and these changes in pulse pressure, and we see that here. So by the time that we reach the arterioles and the capillaries, there is no longer that pulsatile flow that's occurring. So in a condition with arterial stiffness, such that what might happen with increases in age, we have elevations in the arterial stiffness. So you can see that overall these fluctuations in blood pressure are much greater than what we saw over here. And even though those large central arteries are attempting to buffer that increase in pulse pressure, you can see that this pulsatile flow is now reaching the arterioles and also reaching the capillaries. And remember, those capillaries, if we think about the structure of the capillaries, they're extremely delicate, tiny vessels that are not built to handle these increases in pulse pressure and pulsatile flow that might reach them under a condition of elevated arterial stiffness. So this is a figure that illustrates what might happen when this excessive pulsatility that might occur here, what happens when that reaches the perivascular spaces in the brain. So this might lead to white matter hyperintensities, it could lead to microbleeds, it could lead to microstructural damage, as well as impairments in amyloid clearance. In support of this, recent work from our lab has shown that this pulsatility, as measured in the cerebral vessels, is linked to arterial stiffness. So at the bottom, we have arterial stiffness or aortic stiffness. If we're being more specific, we're measuring stiffness in the aorta, and we're doing this using carotid ephemeral pulse wave velocity, or PWV. Some of you may be familiar with this type of measurement. So in both cases, this is pulse wave velocity. And then here, this is the middle cerebral artery. This is the basilar artery in the brain. And this is showing the pulsatility index. So we use 4D flow MRI to get a sense of the pulsatility within that vessel. And then we looked at the associations. So in healthy adults, what we see is that those that have greater aortic stiffness also had significantly higher pulsatility in the middle cerebral artery and the basilar artery. So this was despite the fact that they were healthy, they were cognitively normal, they didn't have cardiovascular disease, and overall the levels of aortic stiffness were quite reasonable. So they did not have excessive levels of pulse wave velocity or aortic stiffness, but we still see an association between those that have higher aortic stiffness and the pulsatility within the brain. So let's talk about arterial stiffness and another factor. So this is data looking at white matter structure. So we initially looked at this using white matter hyperintensities, and this is an image that shows some white matter hyperintensities here. So these are these bright white areas that show up on an MRI scan, and these indicate areas of axonal loss. So areas of the brain that are no longer functioning properly, and so this is linked to cognitive function. So most of the studies that existed that looked at arterial stiffness and white matter hyperintensities, they did so in large epidemiology studies, and so they had elderly adults and they had adults that had a lot of comorbidities such as cardiovascular disease and diabetes and stroke. We looked at these white matter hyperintensities and an index of arterial stiffness in a group of healthy postmenopausal women around the age of 60. So overall, their white matter hyperintensity fraction was quite low, but what we saw was a significant and positive association between those that had a greater arterial stiffness index and a higher white matter hyperintensity fraction, even though they were healthy and cognitively normal. In addition, this association was seen in women who were normotensive and had normal levels of blood pressure. So this is a study that looked at white matter integrity using diffusion tensor imaging, or DTI. What this study showed is that, this is by Suri et al., they looked at white matter integrity, and they looked at pulse wave velocity over a number of years. So they were able to assess the longitudinal change in pulse wave velocity, rather than this study that I'm showing here, which was just a snapshot at a single point in time. So what we're looking at the bottom is this is the change in pulse wave velocity per year, and they looked at this over a span of 4 years. This value here is showing fractional anisotropy, so this gives us a sense of white matter integrity. In this case, a higher level of anisotropy would be considered healthier or better white matter integrity. So what we see is that those adults that had greater increases in pulse wave velocity over the 4 years showed greater reductions in fractional anisotropy. So these two studies, and there are more that I could highlight, but these are two studies that show associations between unfavorable changes in arterial stiffness and reduced white matter health. So what about arterial stiffness and cerebral blood flow, or cerebral perfusion? So this is a study that was also done in Hiro Tanaka's lab, and they investigated middle-aged adults who either had low or high levels of arterial stiffness using pulse wave velocity again. They looked at cerebral perfusion using arterial spin labeling, and we'll talk about some other studies that are using arterial spin labeling in upcoming slides. So what they showed is that those middle-aged adults that had higher arterial stiffness, shown in red, had lower cerebral perfusion in several areas of the brain. Here I'm just showing the figure that shows lower hippocampal perfusion and lower perfusion in the subcortical white matter in those middle-aged adults that have higher arterial stiffness compared with lower arterial stiffness. So this suggests a potential link between the arterial stiffness in the large central arteries and the aorta and cerebral perfusion in the brain. So So this is another study by Surrey et al., and similar to the one that I showed with white matter integrity, that they looked at pulse wave velocity, the changes over a course of 4 years, and they evaluated the cerebral blood flow. So here they have 2 areas of the brain. They have the frontal lobe in green and the parietal lobe in orange. And what we're seeing here is that those adults who had greater increases in arterial stiffness over the 4 years had greater reductions in cerebral blood flow in these 2 areas, the frontal lobe and the parietal lobe. So these unfavorable changes in arterial stiffness are also associated with lower cerebral perfusion. So I'm going to bring this back to a small summary where we talk about the cognitive trajectory using another model. So this is similar to the figure that we looked at before. It's a little bit different setup. So here we have cognitively normal. We can think of this as the latent phase. And then we have mild cognitive impairment, or MCI. That's the prodromal phase of dementia. And then we have dementia itself. And you can see these dotted lines indicating these thresholds. This time, this is showing normal near the bottom and then abnormal near the top. So depending on the biomarker that we're looking at, those change in different directions, but here we're going from a normal to abnormal. So this first line here is brain structure. So we just talked about some factors of brain structure, specifically white matter integrity and white matter hyperintensity. So these changes in brain structure, you can see that those are changing later. So they start to change with when people are cognitively normal, but we see the greatest amount of change during that mild cognitive impairment stage. This line here is showing tau protein. So the toxic protein linked to Alzheimer's disease, you can see that this starts to increase quite a bit during the cognitively normal phase, and then eventually we reach some threshold and where they transition to mild cognitive impairment. And then here we see amyloid. So this is the increases in amyloid, again linked to Alzheimer's disease. So the earlier slide, when we were talking about neuropathology, these 3 factors, amyloid, tau, and brain structure, could be linked in that first slide with thinking about neuropathology. But you can see if we dive into the different biomarkers, there's subtle differences in what changes the earliest. In this case, for neuropathology, the earliest changes are occurring in amyloid. But this line here is showing cerebral blood flow, and this is cerebral blood flow measured by arterial spin labeling. So earlier we just talked about vascular regulation in general. This study is suggesting that in addition to vascular regulation, that cerebral blood flow itself, measured by arterial spin labeling, is one of the first things to change when thinking about going from cognitively normal to mild cognitive impairment. In fact, you can see once we get to the mild cognitive impairment state, you can see that we're already at a very abnormal level of cerebral blood flow. And then once we reach the stage of dementia, everything is already at almost a maximum in terms of abnormality. So again, this highlights the importance of vascular regulation, but specifically cerebral blood flow and cerebral perfusion measured by arterial spin labeling in thinking about the changes that are occurring with age. So one of the goals then is how can we keep cerebral blood flow as normal and as healthy as possible for as long as possible. So let's talk a little bit about how this changes with age. So we just saw in that figure how it changes over the disease phases, but how does it change in normal, healthy aging in cognitively normal adults? Well, as you might expect, with age, it does change, and it changes such that older adults have lower cerebral perfusion when compared with middle-aged adults or young adults. This is a map that shows the results of arterial spin labeling. The areas of yellow are showing higher cerebral blood flow, and red showing a little bit lower cerebral blood flow, so you can see lots of yellow in these young and middle-aged healthy adults. So older adults have significantly lower cerebral perfusion when compared to young and middle-aged adults. But one question that has come up is how this might be different in men and women. We're not going to talk a lot about sex differences or differences between men and women today, but when we think about how things are changing with age, we do have to consider whether or not they are changing similarly between men and women. And cerebral perfusion is one unique example where it doesn't change the same in men and women. So this is results from Liu et al., and this is data from my lab that's unpublished, but they're showing the same results. They're both using arterial spin labeling to look at global cerebral blood flow. We have young subjects compared with older subjects, and then we have men and women. So the women are shown here, and then the women are also shown here. So what we see first off is that women have significantly higher cerebral blood flow when compared with age-matched men. Then when we look at the older age groups, we can see that the older age groups, there's not differences between men and women. However, what we're seeing is the age-associated decline in cerebral blood flow is occurring more so in women when compared with men. So in these studies, it's a mixed group of men and women. In these studies, when we separate them out, when we think about cerebral perfusion, the majority of change with age when adults are healthy and cognitively normal is occurring in women, and there's minimal change shown in men. So this is another study that looked at cerebral perfusion using arterial spin labeling, and they looked at over the age span. You can see that at the bottom of all these is age ranging from 20 to 80. So they used arterial spin labeling and looked at different regions of the brain, and the figures that I'm showing here are all regions of gray matter. So there's a few things here. One is that right off we can see that the authors concluded that women had higher cerebral perfusion at earlier ages in all areas. This is showing the whole brain, and then we have the variety of different areas of the brain. They also concluded that women declined in all regions of the brain, and men only declined in these frontal lobes. So similar to what we saw before, where there wasn't a lot of decline in men globally, this study showed that men did not decline with age in many of the regions or the whole brain, and they only declined in the frontal lobes. But in all these areas, women exhibited a steeper decline in cerebral perfusion compared with men for all regions that they studied. So again, we have a wide age range here, but they did support the previous work looking at the whole brain. Regionally, it does seem that with aging, that women exhibit a steeper decline in cerebral perfusion. So this is a study that was done by my lab as well as Dr. Patrice Broussard's lab and Dr. Sandy Billinger's lab. We got together actually at a small workshop, and we had some questions about the sex differences in cerebral blood flow, and we realized that if we pooled some of our data together, that we might be able to address some of these questions. So in our case, we did not have arterial spin labeling to look at cerebral blood flow, but we did have cerebral blood velocity. So we had cerebral blood velocity in one of the vessels in the brain, and we had this in a number of individuals. And we looked at how the middle cerebral artery blood velocity changed with age and looked to see if it was different between men and women. So this is a study that was done with 529 participants over this age span here. So what we see first, I'm just highlighting the females here. These are where the women are, and then where the men are in this figure. So you can see, similar to what we showed before, that there are some subtle differences between this slope of this decline between men and women. And so we evaluated the rate of change in this slope. So if we looked at the complete slope, we did see differences between men and women. We saw a greater decline in women when compared with men, and we saw that the majority of these changes occurred between the ages of 61 and 70. This is where there was the steepest decline in cerebral blood velocity in women. So this supports some of the earlier work, although we're looking at velocity of blood flow in a single vessel in the brain and still seeing this. The other thing we were interested in was cerebral pulsatility. I mentioned cerebral pulsatility earlier, that we measured it with MRI. In this case, we measured it using transcranial Doppler, and we utilized cerebral pulsatility index to get a sense of how pulsatile the flow is in those cerebral vessels. So again, 529 participants. This is the equation for cerebral pulsatility index. So we look at the peak systolic velocity minus the peak diastolic velocity, and then over the mean velocity within that blood vessel. And so many of the transcranial Doppler devices do this automatically, and it's an interesting measurement of hemodynamics and gives us a sense of how well the vasculature is dampening the pulsatile flow from the heart. So again, this pink line is showing the women, this blue line is showing the men. And so in this case, higher pulsatility index means worse function of the vessel. Ideally, we would want low pulsatility because that means that those cerebral vessels there's not a lot of pulsatility reaching those cerebral vessels. So again, we looked at the rate of change, and overall we did see a significant difference between men and women, such that now women were increasing cerebral pulsatility over the age span that we tested when compared with men. And this time we saw that the greatest difference between men and women was occurring between the ages of 51 and 60. So in both of these studies, that age range after 50, likely during the postmenopause years, are when we see the greatest change in women. Okay, so we talked a lot about cerebral perfusion and changes in brain health and vascular function, but now I want to highlight a few of the studies that looked at the role of higher cardiorespiratory fitness or higher VO2 max or VO2 peak, depending on the study. And I also want to think about what the role of habitual exercise training is. We already saw one study that looked at exercise training on carotid arterial compliance, but we haven't really dove any further into some of the cerebral measurements with the role of exercise. So I'm going back to this multi-site study that we did with Dr. Billinger and Dr. Brassard. We also had participants that had cerebral blood velocity, but also had a VO2 max test. So we were interested in the role of cardiorespiratory fitness in what the cerebral blood velocity was, especially if we look at the differences with different age groups. So again, a multi-site study. This time we only had 153 participants because not everyone had a VO2 max. And their age span for this particular study was 18 to 83 years. So this is looking at the association between VO2 max and middle cerebral artery blood velocity in men and in women. So in women, we saw a significant and positive association such that women who had higher levels of fitness, higher VO2 max, had greater middle cerebral artery velocity. Now, one problem with this is that, as many of you are probably aware, VO2 max changes with age. So we had to statistically adjust for age in this model. And when we statistically adjusted for age in this study, there was no longer any significance. So even in the combined group, in the males or in the females, there was not an association between VO2 max and middle cerebral artery blood velocity if we adjusted for age. We also looked at cerebral pulsatility and the role of cardiorespiratory fitness. So again, same considerations as the slide before, same study, different measurement. This time, we saw a significant association between VO2 max and cerebral pulsatility index. So here, higher VO2 max, higher cardiorespiratory fitness, lower cerebral pulsatility index, and we see this in both men and women. But again, because pulsatility changes with age and VO2 max is linked with age, we had to do some statistical adjustments to account for age. And when we did that, this relationship remains significant, but it remains significant in women only. So in the men, once we accounted for age, no relationship between VO2 max and cerebral pulsatility index. So we found this finding particularly interesting. This was just published in the Journal of Applied Physiology. But really what this suggests, we had a decent sample size, but what this suggests is that we really need future studies to determine if there are sex-specific differences on the impact of fitness on cerebrovascular health. There really are not a lot of studies out there that address this, and certainly not large-scale studies or longitudinal studies that adequately address this. So this is exciting, but it's not conclusive. We need more research and more data to support these findings or to verify these findings. So let's switch gears a little bit. We're going to talk about cerebral perfusion now with arterial spin labeling again. But I brought this figure up again to remind you that the type of exercise or the mode of exercise seems to have an influence at least on carotid arterial compliance. So one of the things that we thought about, knowing what happens with arterial compliance and arterial stiffness, and also knowing the link between arterial stiffness and cerebral perfusion, we wondered if we looked at people who primarily perform running or cycling exercise versus people who perform primarily resistance-based exercise if there would be differences in cerebral perfusion. And to do this, we first looked at young adults only. So these were young adults who had to have years of training in a specific discipline and not do a variety of different types of exercise. So we used arterial spin labeling to look at cerebral perfusion. We had young adults, sedentary controls, aerobic trained, and resistance trained adults. And here's what we found. Now, this is a small-scale study, but nevertheless, in young, healthy, college-age students, these are our sedentary controls, we did see significantly greater cerebral perfusion in the aerobic-trained young adults compared with the control adults. With the resistance-trained adults, this is the p-value comparing the level in the resistance-trained adults to the sedentary controls. So this did not reach significance, and you can see that it's not as high as the aerobic-trained, but it does suggest that resistance training, at least we saw that resistance training did not have significantly lower global cerebral perfusion compared to sedentary controls. So this, again, is a small-scale study. It's exciting to think about, and it was a little bit surprising because usually we think of young, healthy, college-age students as having ideal levels of cerebral perfusion and cerebral blood flow, but here we're showing that even in young, healthy, college-age students that aerobic-trained adults have greater cerebral perfusion than sedentary controls. So there you can see our conclusion based on this. And now we'll move to some data on habitual exercise and middle-aged and older adults. So here we have, this is a study by Dr. Wang Zhang's group at UT Southwestern. So they used arterial spin labeling to look at global cerebral blood flow. And they had three groups. They had a young sedentary group, they had a middle-aged sedentary group, and then they had a middle-aged aerobic trained group. And so here is their cerebral blood flow. These are in specific regions of the brain, so they did show that the middle-aged aerobic trained adults had higher cerebral perfusion compared with the middle-aged sedentary adults, but they also showed that the middle-aged aerobic trained adults had higher perfusion than the young sedentary adults. So this is kind of supporting the study that I just showed you, that the young sedentary adults, even though they're healthy, they can have lower and unfavorably low levels of cerebral perfusion. So this is a study that was done at University of Wisconsin-Madison in a lab in my department from Dr. Dane Cook's group. And so they had VO2 peaks. So these were women who were not aerobic trained, but they did have different levels of physical activity. And so they investigated VO2 peaks, and they split the group of postmenopausal women up based on their VO2 peak. So here's the tertiles of fitness levels, so low fitness, moderate fitness, and high fitness. And then they were looking at a composite score for cerebral blood flow. And what they showed is that those women who had a higher VO2 peak had significantly greater cerebral perfusion when compared with those with a lower VO2 peak. So even here we see in these postmenopausal women, as well as in these middle-aged aerobic trained and middle-aged sedentary adults, that there are differences based on differences in cerebral perfusion based on habitual exercise status or fitness. So these differences in cerebral perfusion are based on habitual exercise and or fitness levels. So we talked a lot about higher cerebral perfusion. So that kind of gets at those two colors of the brain that I showed you earlier, if we have enough cerebral blood flow or we don't have enough cerebral blood flow. But like I mentioned, we think this is linked to the function of the cerebral vessels that are controlling blood flow within the brain, but also controlling blood flow leading to the brain. So one question is whether or not higher cerebral perfusion equals better cerebral vascular function. And so we'll talk a lot about how we measure cerebral vascular function. So one of the difficulties with measuring blood flow in general, but in particular measuring blood flow in the brain, is that it's quite complex. So here is a slide from Phil Ainsley's group where they were looking at, this is a review article, and they're trying to capture all the different things that affect cerebral blood flow. So this is global cerebral blood flow shown here. So this might be similar to the cerebral perfusion that is measured by arterial spin labeling that we talked about in the previous studies. So global cerebral blood flow changes with metabolic activity, it changes with blood pressure or perfusion pressure, it changes with autonomic nerve activity, such as cerebral sympathetic nerve activity, it changes with cardiac output, but it also changes with chemical composition of the blood, so the arterial blood gases. This is showing arterial PCO2, this is showing arterial PO2. So you can see here that the cerebral circulation is highly sensitive to arterial PCO2. So small changes in arterial PCO2, we see large changes in cerebral blood flow. So most often these factors are used to estimate the vasoreactivity or the responsiveness of the cerebral vessels to get an index of cerebrovascular function. So let's talk a little bit more about vascular function in the cerebral vessels. So when we're thinking about perfusion or cerebral blood flow, we have this sort of normal state, the control state, the resting state, whatever you might want to call it. And then we have a certain amount of perfusion pressure that's driving that flow. Then we would expose the participant to hypercapnia, maybe hypoxia or neuronal activity, so increased CO2, decreases in O2, changes in neuronal activity, or cognitive engagement, and this will elicit an increase in cerebral blood flow. Then we can measure this using transcranial Doppler. I mentioned this briefly before. This is showing with each heartbeat the velocity within a single vessel. So the studies that looked at cerebral blood velocity in the middle cerebral artery utilized this approach. We can also use 4D flow MRI, and I'll talk about some studies there. You can see this is showing the velocity vectors within the brain of numerous cerebral vessels. And then other people have used bold imaging. I'll only show one or two studies that utilize bold imaging. So this is blood oxygen level dependent imaging. So it looks at differences in oxygenated versus deoxygenated hemoglobin and gives us a sense of how blood flow is moving through the brain. So these are ways we can capture these changes in cerebral blood flow. And then what we can do, and this you'll remember from the earlier slide, we have cerebral blood flow here. We can change the level of arterial CO2. We can change cognitive demand, and then we'll see a change in cerebral blood flow. So oftentimes the slope of this line is used as an indicator of vasoreactivity, and we can use that value to understand the function of the cerebral vessels. So this is the transcranial Doppler setup. It's kind of hidden here a little bit because we have this mask that one of our undergraduates is modeling, and then we are changing the amount of CO2 and changing what she's breathing, and then we're capturing this using transcranial Doppler. So oftentimes ourselves and other labs might use this to capture the middle cerebral artery, the posterior cerebral artery, the basilar artery, etc. If we're using MRI, this is at the Wisconsin Institute for Medical Research. It's a little bit more challenging. We have large bags filled with different air compositions, and the participant, if they're lying down here, and then they'll be put in the MRI scanner, and they'll breathe those different gas concentrations while we're going through the scans. And that's when we get these really nice velocity vectors through the internal carotid arteries shown here. We have the middle cerebral arteries shown here, and then here is the circle of Willis. So this is an example within the lab using transcranial Doppler to get cerebral blood velocity, just to give you a sense of the other types of data that we collect when we're doing these studies. So this is oxygen saturation shown up here. This is the middle cerebral artery blood velocity. So you can see with every heartbeat the changes in blood velocity. We are measuring end-tidal CO2 to make sure we know the CO2 levels before we change the CO2 levels. We have beat-to-beat blood pressure. We have cardiac output, we have heart rate, and then sometimes, depending on the study, we'll also capture sympathetic nerve activity. Earlier in the slide, I mentioned how cerebral sympathetic nerve activity is linked to changes in cerebral blood flow. This is peripherally measured sympathetic nerve activity, but this could also be a factor that is linked to the regulation of blood flow leading to the brain. So let's talk about some of the studies that are looking at habitual exercise and the cerebral vasoreactivity measurement. So again, I'm bringing up this same slide as before, just to remind you of the differences in carotid arterial compliance based on type of exercise. And we were very interested in this, as I mentioned, so we also looked at cerebral vasoreactivity measurements because these are young, healthy adults, but we wondered if some of these changes that we know occur in carotid arterial compliance might affect cerebral vasculature. So this is looking at the untrained, the aerobically trained, and then the resistance trained young adults, and this is the slope of the relationship between arterial CO2 and cerebral blood velocity. And so what we see, you can see the individual data, we have a lot of variability in the resistance trained and the aerobic trained, but overall the group average were not significantly different. So even though there might be slight differences in carotid arterial compliance, in terms of the cerebral vasoreactivity, we don't see any differences in these groups, at least in young adults. This, if you're interested, this study is also published in the Journal of Applied Physiology, but it's just in young adults, and so this type of work really needs to be followed up in middle-aged and older adults who may have been doing that type of training for decades. So now let's talk about vasoreactivity, but the interaction now with aging and habitual exercise rather than just focusing on the young. So just a reminder of these vasoreactivity measurements, the slope of this relationship that we use. So the first study was just really focused on the effect of age. So this is young and older healthy adults. And when we went back and looked at the data, this data was in sedentary and active. So we didn't have any trained adults that were in the study, but they reported a mix of physical activity levels. So what we see here, much like some of the cerebral perfusion data, that the older healthy adults have lower vasoreactivity than the young adults. We also had VO2 max measurements on these participants. This is showing the older adults only, and this is showing vasoreactivity. So the older adults, there was a significant and positive relationship such that the older adults who had greater levels of fitness had higher cerebral vasoreactivity. So that was an interesting study. We were not expecting to find this, so it's small scale, so we wanted to follow this study up. Even though there seemed to be a link between cardiorespiratory fitness and vasoreactivity, at least in the older adults. So what we did is we initiated a second study where we recruited young and older adults who regularly performed exercise. So they were habitually exercise trained, primarily aerobic trained, not a lot of resistance exercise, and we wanted to see if this age-associated difference in cerebral vasoreactivity was present when we just looked at habitual exercisers. And what we see, similar age range as the study here, but we did not see a significant difference now between the young and the older adults when we looked at the habitual exercisers. So their vasoreactivity was not different between groups. So this is somewhat exciting that there were no age group differences, because that would suggest that perhaps habitual exercise is beneficial for cerebral vasoreactivity. So I've just switched these figures over a bit because I also want to bring up this study from Dr. Rong Zhang's group. So they used a little bit different method of capturing vasoreactivity. They used BOLD, and that means they're also able to get a number of areas of the brain compared to what we were looking at, which was just a single vessel. And they had three groups. They had, this time, master athletes, they had middle-aged sedentary, and then they had young sedentary adults. So the young sedentary adults are shown in green and had some of the highest levels of vasoreactivity. The middle-aged sedentary are shown in red, but the master athletes are shown in blue. And so they actually had the lowest cerebral vasoreactivity in several areas of the brain, such that many of these areas were significantly different between the middle-aged sedentary and the master athletes. So they were actually showing lower vasoreactivity, so poorer cerebrovascular function in the master athletes when compared to the middle-aged sedentary. So therefore, it's unclear how habitual exercise may influence these age group differences in vasoreactivity. We wouldn't want to conclude that exercise is bad because we know that exercise does all these great things for the brain, but this is very intriguing that they saw these types of results for vasoreactivity. So one thing to consider when we're thinking about these cerebrovascular function measurements and cerebral vasoreactivity is the brain is quite complex. When we think about the vasculature in the brain, we have a lot of blood vessels that we're referring to in a very small space. So here's showing the interaction between the capillaries and the neurons. And then the other thing you have to remember is that all of this is encased in the skull with really limited ability to expand and contract, especially if we compare that to, for example, the skeletal muscle in the legs that can readily increase blood flow to the skeletal muscle and not as limited as what we are in the brain. So the cerebral vessels in the brain, the function of them is very difficult to estimate the values as well as understand the function because of the tight coupling and tight regulation of blood flow in the brain. So a couple more things about cerebral blood flow measurement, because we think that this might be why we see some differences with exercise and some differences with aging. There's really not a general or a standard way to capture cerebral vasoreactivity. There's a lot of different methods out there. We're not necessarily saying one is good or one is bad, but there's a lot of different ways to do that out there, and we think that this contributes to some of the variability in the literature. So when we think about cerebral blood flow, and I'm going to focus on the middle cerebral artery because that's what's used most often. This is the one that also for transcranial Doppler is often focused on because it's easier to get with the transcranial Doppler. So we have this middle cerebral artery, and then it branches off into the smaller vessels and eventually the capillaries. So when we expose a participant to changes in arterial blood gases or increases in neuronal activity, the understanding, or our understanding right now, is that these vessels, when we give a vasodilatory stimulus, such as an increase in CO2, that these vessels dilate, and because of that, there's an increase in blood velocity and blood flow through this middle cerebral artery, and our transcranial Doppler is not sensitive enough to capture this, so it's really just focusing on the blood velocity right here in the larger artery. So one of the problems is that it has been suggested that the middle cerebral artery may also change in diameter when we give some of these vasodilatory stimuli, so that if that's the case, then this measurement of blood velocity may not be ideal to capture what's happening with cerebral blood flow. So we sought out to test this a little bit more, and we compared two methods of capturing cerebral blood flow during a CO2 challenge. So this was transcranial Doppler ultrasound, and this was the 40 flow phase contrast MRI. You can see the different methods here. We did focus specifically on the left middle cerebral artery so that we could compare compare the same vessel directly. So in this study, we had 40 participants. They completed both study days in a randomized order. And what we found, these are all healthy habitual exercisers. If we looked at their whole brain or the middle cerebral artery in the young and older adults, you can see that there's a trend maybe for the older adults to have slightly lower cerebral vasoreactivity, but there was no statistical differences between these groups. When we looked at the differences between young women and young men, and then older women and older men, we again did not see any differences based on age or based on sex or based on the interaction. Now, when these same participants came into the MRI scanner and we focused on the middle cerebral artery, so here, up here, there's no age, sex, or interaction effects. Down here, when we brought them in, same participants, now we see lower vasoreactivity in the older adults. We also see that there are sex differences such that young men had higher vasoreactivity than young women. We see that the age effect seems to be driven primarily in the men in the case of cerebral vasoreactivity. And we don't see any differences between young women and older women. So if we compare this figure here and this figure here, really what we see is this one seems to be amplified, so that when we have more information, when we have information on velocity and cross-sectional area of that vessel, the potential differences between groups seems to be quite a bit larger than when we just focus on cerebral blood velocity by itself. So we do see some sex-specific effects using 40 flow MRI, primarily these differences between men and women in the vasoreactivity measurement. So let's look a little bit closer at this vasoreactivity measurement using MRI. So this is the cerebral vasoreactivity. Now, in addition to just the left middle cerebral artery, you can see that it's the same trend when we look at the basilar artery and when we look at the left and the right internal carotid artery. So similar trend. It's not that one area of the brain is changing differently than another area of the brain. So this left middle cerebral artery is what we focused on in the previous slide because that's what we were able to get with the transcranial Doppler. So one of the things that we were thinking about is why might these be different? So we decided to look a little bit closer at this left middle cerebral artery and look at how the cross-sectional area of that artery changed when we gave carbon dioxide to do this vasoreactivity measurement. So you can see the change in end tidal CO2 here, and then the cross-sectional area. So each of these gray lines are the individual data, and then we have the group averages. So you can see that the men had had actually significant dilation in response to CO2 compared with the women. The women, there was no significant difference, but you can see a lot of variability amongst these individuals. So this really highlighted the idea that cerebral artery dilation might be a measure of cerebrovascular function and a way to assess how the cerebral vessels are doing. And so we looked at this a little bit more. So we do this measurement during our vasoreactivity measurement because we give CO2 and we're able to look at cerebral artery dilation. And this is the data that I'm showing on the young and the older adults. So you can see the two groups here. The dark black line is showing the group average. So right away, what jumps out is that the young adults, you can see the dilation, but the older adults, there is a lot of variability in the dilation of that left middle cerebral artery. So we also determined that there was significant cerebral artery dilation and greater blood flow in the young adults. So overall, this greater cross-sectional area increase. Now, this is a mix of the men and the women here, but overall, when we combine them, there was a significant dilation. We did not see significant dilation in the older adults. But really, what this highlights is the variability of the response, particularly in these older adults. So one interesting thing is we had some data because we wondered what some of this variability might be, what might be caused by. So this is looking at VO2 max data. So this is VO2 max here, and this is the magnitude of change in cross-sectional area of the left middle cerebral artery. So we didn't have this in all 40 participants because we didn't have VO2 max on everybody, but what you can see is a significant and positive association such that those that had greater levels of fitness also exhibited a greater change in left middle cerebral artery cross-sectional area. So this was exciting and interesting. And when we looked a little bit further, it wasn't just in VO2 max, it was also in maximum workload, as well as exercise volume that was self-reported, was also significantly associated with the middle cerebral artery cross-sectional area change. So now we've become very interested in this cross-sectional area change and how that might relate to exercise training or fitness, and if that's something that exercise is able to initiate an adaptation. Now again, this was a small study. This was probably not adequately powered to test this really well, so we need to do follow-up studies, and we have follow-up studies planned to investigate this further. But this is linked, or it is supported by some of the data in the animal literature. So this is comparing sedentary animals, shown in the white bars, compared to animals, these were all done in Sprague Valley rats, that underwent vigorous exercise training, and those are in the black bars. So this is in response, this is the change in diameter of the middle cerebral artery in response to concentrations of ADP. So a vasodilatory stimulus at 2 different concentrations. And what we see here is that those rats that underwent vigorous exercise training had significantly greater changes in diameter relative to the sedentary controls. So this would support this, what we found in the older adults, that there might be something specific about vigorous exercise training that may initiate an adaptation in the cerebral vessels and increase the ability to dilate the cerebral vessels. This is a follow-up study from the same group by Eric et al. This is also in animals, so they also investigated sex differences. Again, we're looking at the concentration of ADP, the change in diameter of the middle cerebral artery, but now we have a few more groups. So the white ones are the male animals, the darker bars are the female animals, but now they have the exercise training in there as well. So what we see in both cases is that here the females had greater change in diameter relative to the males, but we don't see any differences between the hatched bars and the solid bars, and that's the moderate exercise train group. So, for example, if we focus on this one here, these are the male sedentary animals. These are the male animals who underwent moderate exercise training. These are the female sedentary animals. These are the female ones who underwent moderate exercise training. So even in the same laboratory, they're not seeing the effect of exercise training in this particular study using moderate exercise, but they did see this difference when they looked at, or they had animals that underwent vigorous exercise training. So lastly, I'd like to wrap up with some data on exercise interventions. This is not an exhaustive list of exercise interventions, but I did want to give you a sense of some of the data that is out there. So this is the results of studies that looked at exercise training on cerebral blood flow or cerebral blood velocity or vasoreactivity measurements. So first, we have some studies that showed that an exercise training intervention in older adults had no effect on cerebral blood flow. And so I have two studies that are listed here. One used 16 weeks of moderate to vigorous exercise. One used 6 months of low to moderate exercise. In contrast, we have some studies that have shown an increase in cerebral blood flow with exercise training in older adults. So we have several studies that were 8 to 12 weeks and used moderate to vigorous exercise, and they also used a progressive intervention, so increasing the exercise dose throughout the intervention. And we also had another 6-month study that had moderate and progressive exercise that did show an increase in cerebral blood flow. And there are some studies in the literature that show that exercise training has no effect on vasoreactivity, also moderate and progressive exercise. And we've also seen some that have shown an increase in cerebral vasoreactivity with an exercise training intervention. This one was 12 weeks and moderate to vigorous. So I realize that these are showing no effect or increases, but I just wanted to let you know that there is a lot of literature out there in exercise interventions, but the data is really mixed. And the other thing to remember is that some of the methodology in which we use to capture cerebral blood flow and cerebral vasoreactivity has been improving over the last 15 years. And so some of these studies were done many years ago. Some of these studies are more recent. And so we have not just the effect of exercise, but we also have some changes in what methodology was used to capture these measurements. So the question that this brings up to me is whether or not there's a threshold dose of exercise necessary to increase cerebral blood flow or vasoreactivity. The studies that I've selected here, and again, there are many more studies out there, I try to be very selective for those who that conducted exercise in a very structured manner, consistent with exercise guidelines. So we don't know, though, if there's a certain dose necessary. We know what the guidelines recommend for cardiometabolic health, but whether or not that dose of exercise is necessary, or if a greater dose or a lower dose would be required for changes in cerebral blood flow or vasoreactivity, we don't yet know that. So I'll end with data from Dr. Ron Zhang's group on aerobic exercise training, and these were in mild cognitive impaired patients. So I wanted to highlight the study, one, because it's promising, two, because it shows how some of these things might be linked, and also it's a very unique and well-conducted study, and there's not a lot of data out there currently with exercise interventions in MCI patients. So this is some information about the intervention. It was 1 year. It was moderate to vigorous intensity. It was progressive. And they also included high-intensity exercise sessions. And importantly, they demonstrated an increase in VO2 peak. So this is the intervention group that did active control group, and this was the aerobic exercise train group. And so you can see that they had significantly greater increase in VO2 peak in this aerobic train group relative to the control group. This is showing that stretching active control group. This is showing cerebral blood flow. This is showing carotid arterial stiffness. So what we can see from baseline 6 months and 12 months is there's really no change and no difference in these variables. Again, cerebral blood flow and arterial stiffness. Here is the aerobic training group. So they did show significant increases in cerebral blood flow over the course of the intervention, which is exciting. They also showed significantly lower or decreases in carotid arterial compliance over the course of the intervention. They also showed that the magnitude of change in VO2 peak, so that's shown here, was linked to both cerebral blood flow, shown here, and arterial stiffness. So those that had the greatest increase in VO2 peak showed the greatest increase in cerebral blood flow, and also showed the greatest decrease in carotid arterial stiffness. So this really highlights the linking between what's happening in the large central arteries and what's happening in the brain, but it also gives us a sense of how important the change in VO2 max, or the intensity of exercise that was used, how important that might be to these results. And finally, they also looked at white matter integrity. So they looked at mean diffusivity, so it's a little bit different measurement than what we talked about before, but still gives us an assessment of white matter integrity. And they looked at this with the intervention. So this is showing, similar to the figures that we just looked at, this is showing the change in VO2 peak. So in this case, the mean diffusivity, so the lower value, would indicate better white matter integrity. So in all of these, I'm just showing 4 regions right now. In all of these regions, those MCI patients who demonstrated a greater increase in VO2 peak demonstrated the greatest reduction in mean diffusivity. So the interpretation would be the greatest increase in VO2 max, the better the white matter integrity over the course of the intervention. So this exercise intervention demonstrates that improvements in VO2 peak and the reductions in arterial stiffness had a beneficial effect on both cerebral blood flow and white matter integrity. And this is a very promising study, and I know that they have a lot of follow-up studies planned, and I look forward to hearing the results of those studies. So with that, I'd like to summarize what we talked about today. We think that the function of the large central arteries, the function of the aorta, and the function of the blood vessels that lead to the brain, as well as the blood vessels in the brain, are very important for thinking about brain health. We showed some data linking the function of these vessels to cerebral perfusion. We looked at the linkage to vasoreactivity, and we also looked at the linkage to white matter integrity. Now, we think that one way that exercise may potentially affect these measurements of brain function here are because the regular vigorous exercise has an impact on the vascular function measurements. And so notice that I specifically said vigorous exercise because both the human data as well as the animal data would suggest that the vigorous exercise and or a change in VO2 max, that might be a necessity to see adaptations in both the vasculature as well as in the brain. But I also want to add that there's still a lot of questions that remain. Some of the questions are about the measurements, which measurements we're using, the technique that we're using to capture it. A lot of the studies looked at vasoreactivity to arterial CO2. There's other ways that we might be able to do this. I also mentioned the cerebral dilation might be an interesting measurement that we're interested in. We did not even touch on potential underlying mechanisms. There's a lot of unknowns about the underlying mechanisms, and we often have to rely on the preclinical and animal data, animal literature, to help us understand that. And then another question is about exercise dose. We know that there's some ongoing trials that are going to hopefully address the idea of exercise dose, of thinking about optimizing the dose of exercise necessary to get beneficial changes in the brain. And we also look forward to some of those results coming out. So with that, I'd like to acknowledge the collaborators for the studies that I presented today, as well as the past and present trainees who assisted with this work. I'd like to acknowledge our funding sources for the studies that I presented today. And then here's a picture of my lab. We're the part of the Bruno Balke Biodynamics Lab at the University of Wisconsin. You can follow us here on Twitter. And with that, I thank you for your attention. And I wish I could be there to take questions, but I hope that you have a wonderful conference, and I hope to meet some of you in person soon.